So you mix up the cells, you imagine them kind of floating around in this stuff. You pour that liquid into a mold, and over a couple of hours that mold will kind of firm up and form a simplified tissue. And by guiding that process, you can create very sophisticated tissue structures that mimic the natural tissues.
Kevin Costa of Mount Sinai is describing the process of coaxing stem cell-derived heart cells into functional human heart muscle. Mount Sinai is one of the few institutions in the country that has been successful in engineering a 3D human tissue model to study heart disease.
It's really difficult to have access to natural human heart muscle, and so our idea was to make our own and make something that could be a replacement for natural heart tissue.
These heart tissues are created from human stem cells able to beat 60 beats per minute, much like the human heart.
You treat the tissue kind of like you would treat the patient, right? So if the patient needs a drug to make their heart beat more strongly, you can test that drug on the tissue and see if it makes the tissue beat more strongly. And being able to test drugs earlier on in the development process on actual human heart muscle increases the chances, we think, that you'll be able to exclude the ones that aren't going to be promising and focus on the ones that really look exciting.
Scientists say that using this screening approach in engineered human heart muscle allows the possibility of overcoming some limitations of animal model studies, enabling science to move more efficiently from bench to bedside.
Our major research effort now is looking into how we can accelerate the aging and maturation process to go from something that behaves like a newborn heart muscle to something that behaves like an adult heart muscle, because most heart disease, at least acquired diseases, occur during adulthood.
The technology being developed at Mount Sinai is part of a new era of precision medicine, where engineered tissues created from a patient's own cells might be used to help identify possible drug side effects and select the best treatment for that individual.
We really think that it would be wonderful to be able to take this technology and make patient-specific heart muscle that we can then pre-evaluate different drug therapies and inform the cardiologist or inform the physicians that's treating those patients as to what's going to be the most effective therapy for that patient.